little children, I welcome you all to PU2 Brahmastra series. Today is day 52, and first things first, kudos to all of you who were with me throughout 13 chapters, my dear children. We have finally completed biology. Now this video is for you to understand what diagrams are important, okay, which diagrams would come in the exam for you to draw, which diagrams are just important for 1 marker question probably because the labelings of it might be asked but it might not come for drawing, okay, or probably the line underneath the picture is very much important. So I am going to tell you all about that, and I have divided the diagram video into 2 parts. So I hope you understand the importance of it. Even 5 marker questions, 3 marker questions have come from the diagram aspect of view, so you should be knowing the diagrams really well. Yeah, so kudos to all my dearest and cutest little children. I am really proud of you, and let me tell you children, I am not a person who draws really well, okay. My drawing, I call myself as a failed Picasso. It's a fun thing that I like to denote myself like that. But anyways, I hope that lots of you can relate, okay. You also might not be that great at drawing. So yes, so people who relate with me, you can definitely comment it down in the comment section. But today we are going to practice few diagrams, and I am pretty sure you will be remembering all of them. Yeah, chalo, let's get started. So how to remember the diagrams? Before getting into that, let me just talk about a small thing that is our sister channel, a cute baby sister channel. Okay, so we have Diksha Vedantu Foundation channel as well, which is primarily for 8, 9, 10, but we have focused on 10th only this year because it has just started off, and 10th has board examination. So just like you guys, they also have board examination, they also have to prepare. So if you or someone who have siblings studying in 10th grade, you should definitely recommend them this channel. We have been putting our efforts in there as well. Okay, so it's the same set of teachers who are there. Okay, so definitely do not forget to subscribe to a cute little channel that is out there. Now, the right way to remember diagrams. First things first, you need to understand the diagram. Okay, without understanding the diagram, you are not going to remember anything. So you should be knowing that, okay, for example, if you talk about the male reproductive system, you should be knowing that there is the testis where the sperms are being made. Now when you know that, probably you will be making the cells that will give rise to sperms in the testis itself. And if you know that acidic ducts are the ones that transport the sperm, then probably you will be transporting the sperms from the testis to some other organ. So if you know the function, you would be able to relate and learn it. So understand the diagram really well. Now once you have understood everything, what do? You do the first trial. And how do you do the first trial? You look at the picture and you try to draw. You look at the picture and you try to draw. Okay, that is the first thing. Second time when you are going to draw without reference, my dear children. Close the book and try to draw it. Okay, close the book and you try to draw it. Now suppose you have drawn something, you drew something like this, where this is a testis and this is the vas deferens, and it is going around here. You have your, what do you say? This is the urinary bladder and everything. Okay, it looks like a cup. Anyways, okay, I'm not wanting to make it perfect. So you made a gland over here and you did something like this. Now this is what you drew without what? Reference. Now what you have to do is open your book and try to label it. So now when you label it, you get to understand, okay, this is testis, fine, and this is vas deferens, and this is seminal vesicle. Oh, but where is the prostate? Prostate is not there. So I have to draw the prostate as well. That is the use. So when you have the picture with you and now you're looking at the labelings and you're finding that this thing is not there in my picture, you will probably draw it. This is how you will remember things better, and you will label it. And you will label it while seeing it. Okay, so your picture thing is clear. Your picture thing is clear, okay, but labeling, you saw and you labeled it. Some of the things you might be able to remember it as well. Now, you have to draw the picture a third time as well. In the third time, you are drawing it perfectly. Okay, in the third time, we will definitely draw it perfectly because we have enough amount of experience as of now. Now, what I want you to do is, after having the image being drawn perfectly, what you need to do is, you have to keep it unlabeled. Do not label that particular image. Label it after 2 days. Now when you label it after 2 days, you would be recollecting things. You will be you will think about it and then you will label it. And if at all you are not getting, then take the use of the reference that is your NCERT textbook. So this is how your labelings and your diagrams will be really strong. Works? I hope it helps. If these tips really help you well, definitely let me know in the comment section. I would be really happy to see that. Chalo. Now, let's starting from the chapter that has the highest weightage, that is 13 marks, Principles of Inheritance and Variation. Over here, this picture would not be coming for you to draw. But what will this picture offer you, my dear children? This picture will offer you questions on what are the features that were selected by Mendel for his pea experiment. Why pea plant was selected? Because there were 7 pair of contrasting traits. 7 pair of contrasting traits, my dear children. So you should be knowing what are those contrasting traits. So over here you have seed shape, seed color, flower color, pod shape, pod color, flower position and stem height. So what you can do is, if you are not able to remember it, you can put the flower position here. So seed, seed, flower, flower, pod, and then one is what? Stem height. Gives you 7 pairs. So this picture is important because of this. They might ask you 7 pair of contrasting traits, you should be knowing it. If you have an image reference, it becomes all the more clear for you to remember it better. Okay? These are the 14 characters or 7 pairs of characters, either way. Now over here, this picture again can be given in the MCQ. They can ask you, what does this particular thing represent? They have given you a box, they colored it, and they are asking you, what does this represent? Is it affected male? Is it affected female? Is it non-affected male? Is it non-affected female? You should be knowing it. Whenever that thing that is, square comes, that means you're talking about male. And now, when it is colored completely, you're talking about affected male, my dear children. The circle, it stands for female. Whenever a structure like this comes, that means the sex is not being specified over here. So whenever these things come, whenever it is shaded, understand it is talking about affected individuals. So what do you do? What do you do with the NCERT? Go put a tick mark on both these pictures, because both these pictures would not be asked for you to draw. Instead, the questions could be coming on the basis of this particular pictures. Okay, you don't have to draw it, but questions would be coming. So this picture is really important. Now what does this line show? That means mating is happening. When 2 lines are there, that means mating between relatives is happening, or you call it consanguineous mating. Okay. Now here, what do you mean? A male and a female mated, and they have a male child and a female child. That is what is being shown over here. A male and a female is have mated, and what do you get? An affected son. What do you mean by this? I'll zoom it in and I'll show you. This means 5 of them, 5 unaffected because it is not shaded. And do you have the sex being specified, my dear children? It is not specified over here. So there is 5 unaffected offspring being produced over here. Works? So I hope you have understood it, what type of questions would also come from this, and this is an important image. Now let's move on to the next one. Ayo. Just a second, okay. Okay, this is on page number 72 and this is on page number 54. Go tick. Right now, when I'm talking to you, you have to put, do not keep it for the end. So I hope you have NCERT. Pause the video, go put a tick and come back. Cool? Next video. Next picture. Sometimes I just talk free stuffs. Okay, so over here, again you have a picture. Let's see on which page number as well. Where is my clicker? Yes, so you have it on page number 73, my dear children. So there is 2 pedigree charts given over here. Okay, you might be given the pedigree chart as such, or any other pedigree chart would be given, and you would be asked which type of disease does it show. So how do you understand which type of disease? Basically over here, mostly they give autosomal dominant disease or autosomal recessive. Okay, so I hope you know that autosomal dominant, as in, for example, I have something like this. In both these cases, since dominant alleles are there, this person and this person, both of them will be affected. Instead, if this is the condition, they will be not affected. This is what you mean by autosomal autosomal dominant. Autosomes as in the autosomal chromosomes, they are affected over here, okay. This is autosomal dominant disease. What do you mean by autosomal recessive? Autosomal recessive means in this condition it won't be affected because there is a dominant allele over here. But when condition arises like this, that particular individual who is having something like this will be affected. This is what? Autosomal recessive. So let's see and understand better. So over here I have a female who is affected and a male. They have this many number of progeny. I see that this guy and this girl is affected. How do I understand whether it is autosomal dominant or recessive? My dear children, if probably it was autosomal recessive, okay, I might not have seen it over here. This particular disease, I might not have seen it over here. Okay? Or basically I can make you understand things better. Over here, it is given that again, their progeny also have this particular disease. That means it is a dominant trait. Okay, it is a dominant, let's say autosomal dominant disease. How do I identify that? Suppose, if it was capital R smaller and this person has smaller, my dear children. This is capital R, which is why this particular person has the disease. Now, when fusion happens, it is capital R smaller and smaller, right? The gametes will be capital R smaller. Now, when you want to fuse, when you fuse this and this, you get capital R smaller. Sorry, not this and this. My God, what am I doing? Okay, when I fuse this and this, I get capital R smaller. When I fuse this and this, again I get capital R smaller. When I fuse this with this, I get smaller. When I fuse this with this, I get smaller. So I have a possibility of organisms being like this. I have a possibility of organisms being like this. Probably both of them are capital R smaller. And what about all of them? They are smaller. Okay, got it? Now again, when both of them, they mate, one among them, or basically any one among them can be like this. That is the one over here. Cool? Works? So dominant trait in every generation it will be seen. If it is an autosomal dominant disease, it will be seen in every generation, my dear children. So this generation it is there, their child also have this, their child also have this particular disease. That is what you mean by autosomal dominant diseases. Whereas when you talk about autosomal recessive ones, if you see over here, okay, if we see over here, the parents do not show the disease. The parents are not showing the disease at all. How can that be possible? If the parents' genotype is like this, both has capital R, it should be diseased if it was dominant. But it is not dominant, which is why both the parents are not going to show the character over here. That is the disease over here. So both of them, they are carriers. They are what? Just the carriers. Now suppose these carriers, that is capital R smaller and capital R smaller, they decide to fuse. So you have capital R smaller and capital R smaller. This fuses with this, you get capital R. This fuses with this, you get capital R smaller. This fuses with this, you get capital R. This fuses with this, you get smaller, my dear children. In that case, over here, you see, if it was dominant disease, then, okay, if we assume that these guys should also be having, then here the parent should also have this if it is a dominant disease. But I don't think it's a dominant disease, otherwise they would have had it. Over here, these things that you see is having smaller, which is why, okay, which is why they are getting affected. Again, these guys which are up over here, probably they are like this. Which is why they are not affected. Okay, they might be like this because there's a dominant allele to save it. Now, again, when they fuse, when this capital R smaller and this capital R smaller will be fusing, you get an offspring which has smaller. Please remember. So, in the case of autosomal recessive ones, okay, you are not seeing in every generation. Okay, you are not going to see it in every generation, and the parents might be just the carriers, they might not be affected, but the child will definitely have it. In the case of dominant one, the parents will definitely, one of the parents will definitely have it. Over here, the parents can be just carriers, my dear children, please remember it. If it was a sex-linked disease, then probably the sexes would be affected. Okay, for example, the male, then only male will be affected, or female, then only basically sex-linked disease, or works like that. Anyways, if you just remember at least this, you might be able to score better. Okay, and here one more example is given. Autosomal recessive trait shown by sickle cell anemia, my dear children, and autosomal dominant trait is shown by myotonic dystrophy. In this particular disease, basically this particular disease is caused by, because it is an autosomal dominant trait. Okay, myotonic dystrophy is an example which is just given in this particular picture, nowhere else. So that's why I am telling you, have proper understanding. Okay. Myotonic dystrophy is an example of autosomal dominant trait. Okay, whereas sickle cell anemia is an example of autosomal recessive trait. Page number 73, again very important. Okay, going ahead with the next thing that we have is the next chapter, that is molecular basis of inheritance, my dear children. Over here you have the structure of nucleosome. Why is it not working? Okay. You have the structure of nucleosome given on page number 83. You have to draw it. Okay, you have to draw it. Make one big circle. This big circle that you see over here is made up of 8 histone proteins. How many? 8 histone proteins. You call it an octamer of histone, my dear children. And on this histone protein, what do you have wrapped around? You have the DNA being wrapped around. Okay, now this is one nucleosome. This whole thing that you see is one nucleosome. Similarly, you would be having another one also. And between two nucleosomes, you see this one histone protein. This is a separate histone protein. This is known as H1 histone, my dear children. You have to draw it. So what do you do when you have to draw it? You put a star mark. This question comes. So usually it is like this, where you have 1, 2, 3, 4, 5, 6, and the 7, 8. Okay, it's in back side. Now over here, you have to rotate basically the DNA twice. The DNA rotates twice around it, and then, okay, then again what do you have, my dear children? You again have what? An histone protein. Then again the same structure being repeated. Okay, so what you can do is just make a round and just show like this. That works. Okay, and then again make an histone protein up over here. It is more than enough. Fine. So one is going back side and the other one comes front side. Fine. Now, translation process. This question has come. This is on page number, okay, let's cover page number 92 first. So please put a stop mark over here. Let's go to page number 92. You have to draw this as well. They ask you to draw a transcription unit. What is there in a transcription unit? Very simple. There will be a strand like this. This is a template strand in 3' to 5'. Okay, basically here it's 3', there it is 5'. Okay, the arrow goes like this. And again you have a coding strand going from 5' to 3'. Initially you will be having a promoter sequence which would help in the promotion of transcription. Okay, the row factor basically comes and attaches over here. Okay, not the row factor, basically the factor, initiation factor comes and attaches over here and start off the process. Then you have a structural gene and then you have the terminator region. You have to know it. In just a second, my colleague has come here.
Can I take your phone?
My phone?
Recording a video. Answers.
Yeah.
No, it's fine.
Okay. You will delete it. This portion. Yeah, so terminator region is there, okay, at that end, that is this particular area. And what do you have over here? You have the structural gene. So this picture you have to draw. This is a transcription unit. Over here RNA polymerase comes and attaches, okay, and then basically begins transcription in this particular direction, that is 5:3 direction. So it will come, take this strand and then make a new strand up over here. What strand? RNA strand. Works. This picture has come, might come, so please practice it. Now translation, this you do not have to draw, but they will ask you questions on the basis of this particular image. I'll tell you what type of questions are being framed up over here. So for that I have to zoom it in a bit. So this is a ribosome unit. This is a larger subunit, and this green one smaller subunit. Okay, this thing that you see is an mRNA molecule. See the direction, very much important. This is 5' and the other one is 3'. Okay, this is 5' my dear children, and the other one is 3'. It is read in this way here. Okay, it is read in this way up over here. Cool. Now they will give you X over here this side and Y on that side. So you should be knowing that this is 5' and this is what? 3' my dear children. Works? Now over here they will ask you if this is the codon up here in GUU, okay, or in the case of valine, what is there in the anticodon loop of the tRNA? Anticodon loop as in you are talking about this loop. So if it is GUU up here, then it is CAA. This is the codon. These are the anticodons. The tRNA always have anticodons, and this region is what? The codon. Okay, or basically these things are codons, which comes in 3. Okay, now they might ask you a question where they can give you something like this. Okay, instead of UUC, they can give you UGA. Okay, they give you UGA or UAG. Let's take UAG. UAG. They'll ask you, will the tRNA come and attach over here in UAG? No, it does not. Because why? It is a stop codon, my dear children. UAG, UAA, okay, UAG, UAA, UGA, everything basically they are what? Stop codons. They are what? Stop codons. So tRNA would not come and bind over there. It will be free. Please remember that. I am talking about the codons, not the anticodons. Okay. So that is the question. That type of questions can be asked from this particular area. So this and this you have to practice it really, really well. Going ahead. Again, something that you have to draw. That is the lac operon. Okay. In lac operon, you have this gene, that is the regulatory gene. Okay, which is going to code for the regulatory protein. It can be a repressor protein or it can be an activator protein. Here it is a repressor protein in the case of lac operon. And this is each gene has a promoter sequence to which the RNA polymerase comes and attaches. Okay, so here also it also has a promoter region identified by P. So PI, okay, PI for inhibiting gene, that is what it means. Okay, now over here this is again another gene. This thing that you see over here is another gene. This is the structural part of this gene, and P is what? P is the promoter sequence. P is what? The promoter sequence onto which the RNA polymerase comes and attaches. I told you, right? What is O over here? O is operator region, my dear children. O is operator. Okay. And Z, Y and A. Zia. Okay. Z, Y and A. Each of them, they are genes coding for proteins. Okay, so Z gene codes for beta galactosidase, Y gene codes for permease, and A gene codes for transacetylase. Fine. Now, if you want to make this proteins, then the operator region should be free. It should not be covered by anything. But suppose the operator region is covered by a protein, what will happen? The RNA polymerase which comes and binds over here and basically helps in transcription, it won't be able to transcribe. It just gets stuck over here. That is the content. Okay, and what is that thing that is coming and going to bind over here? The protein made by this inhibitory gene. Okay, that is a repressor protein, my dear children. This repressor protein goes and binds there. So how do you remember? PIPOZYA. This is the sequence. PIP, Piposia. Okay, Piposia, or whatever or however way you want to remember it, you can remember it. But this will be coming in drawing. If you have lac operon question, you should be knowing it. Okay, so in the absence of inducer, what is the inducer? Lactose is the inducer. So when lactose is there, what it says? Lactose or allolactose, when it is there, it goes and binds to the repressor. If repressor is bound already, it won't be free to come and bind over here. If it is free, this particular region is free, transcription will happen and all these enzymes would be made. So in the presence of inducer, this is the case. In the absence of inducer, this is the case. Okay. So molecular basis of inheritance, these are the things that are really, really important. Lac operon on page number 101. Cool. Put a star mark there. Now one more thing is there. Okay. Page number 98, tRNA. So tRNA, you should be knowing on what side basically amino acid comes and attaches. So tRNA basically it also has 5' end and 3' end. It's on the 3' end that the amino acid comes and attaches, and this is known as amino acetylation, or basically charging of amino acid. That is what you mean by it. So basically charging of tRNA, okay, charging of tRNA or amino acetylation. So over here the serine is coming and binding over here at 3' end. Now this loop that you see is an anti-codon loop which will be going and binding on the codon. So these things are really important. If you are given the structure of tRNA, where does amino acid come and bind, what is this particular region known as? This is known as anti-codon loop. And what is this process known as? Amino acetylation or charging of tRNA. Cool? Done? Fine. So that chapter is also completed, that is molecular basis of inheritance. Now you have what? Human health and diseases. In human health and diseases, the stages of the plasmodium given on page number 132 is really important for you to understand the concepts. So you should be knowing that, okay, so over here what is happening? Mature infective stages, sporozoite. So with this you get one MCQ. What is the infective stage in the case of malaria? It is the sporozoite stage, which is the infective stage of this particular disease, that is malaria. Okay, sporozoite stage of what? Plasmodium, my dear children. So sporozoite escape from the gut and migrate to mosquito's salivary gland. Then when the mosquito bites another human, sporozoites are injected with bite. So they inject sporozoite within us. Now this parasite reach the liver. So first stop is what? The liver, my dear children. From the liver, the parasite reproduces asexually in the liver, bursting the cell and releasing into the blood. So there they reproduce asexually, then they get into the blood. In the blood, where children? RBC. Now, parasites reproduce asexually in red blood cells, bursting the red blood cells and I think I'm hungry saying bread and all, okay, and causing cycles of fever and other symptoms. So now you know why you get fever and other symptoms. Why? Because the RBC is getting broken down, and at this particular point of time, hemozoin, a protein is being released over there, okay, hemozoin, which is why you get fever and chills. Now, sexual stages, that is gametocytes, develop in the RBC. This is very important question. Where does these sexual stages form? In the case of basically the plasmodium. So you should be knowing it is forming in where? In the RBC itself. Sexual stages, that is gametocytes. Until now they were doing asexual. Now they are doing sexual mode of reproduction. They have not started doing, I am just saying they have reached the stage. They will do sexual reproduction only inside the mosquito. Please remember that. Okay, so they are ready and they have formed the gametocytes. Now these gametocytes are taken up from the blood with the help of the mosquito. In the mosquito's gut, these gametocytes are going to give rise to gametes, and these gametes are going to fuse and fertilization, everything will happen. Again they will form some stage, this stage will transform into another stage and now reach sporozoite stage. The plasmodium will reach a stage that is known as sporozoite stage, in which in this stage they are finally in the salivary gland. So they were in the gut, the mosquito came and drank it, okay, they drank it completely. Now the gametocytes are inside the gut. The gametocytes are going to form gametes, the gametes are going to fuse in there, and now ookinete and things like that are going to form. These things are going to move up, okay, into the salivary glands, they will form something else, and this something else is what? Sporozoite, my dear children. Okay, this something else is sporozoite. So you should be knowing that the sporozoite which is formed in the gut is going to migrate into the mosquito salivary gland, just going to stay there, and when it comes and bites us, it is going to give us that. Cool. So you have got almost 3 questions coming off from this particular picture itself. Okay, the gametocytes and everything is not given in the theory part. So you should be knowing about it. That's why I am saying this picture is really important. So what do? You put a tick mark over there. Again, this picture that is given on 138 page number, that is about lymphoid organs. Not all the organs are mentioned over here, but they can ask you, they won't give you thymus, they will ask you label it. So you should understand the one that is in the middle, in the chest region, it is always the thymus. One that is over here is thyroid. Okay, so thyroid is not a lymphoid organ, thymus is. So you should be, thymus is a lymphoid organ which is over here. Over here you have tonsils also. Tonsils is a lymphoid organ. That comes a separate thing. Okay, so thymus is a primary lymphoid organ which is present in the middle. Please remember. And these node-like structure, the purple colored thing that you see over here, node, okay, round things, they are known as the lymph nodes. Cool? Chalo. I hope you have marked it. Put a tick mark. Okay, put a tick mark. and mark it important. Mark this as important. Mark this as important. Cool? Chalo. Next thing, structure of an antibody molecule, very, very, very important. Every time they ask this, page number 135. Okay, so what do you need to draw? You need to draw a heavy chain first. Fine, this is the heavy chain. Heavy chain will have a region which is known as variable region. What do you call it? Variable. It is different in different chains. Okay. Now there is light chain as well. This light chain also has variable region. Similarly over here also they have what? Variable region. Light chain also has variable region. What is the rest of the region known as? It is known as constant region. It is same in all type of antibodies. Constant region, my dear children. So two heavy chains, which is why I call it H2, and two light chains, which is why I call it L2. Okay. Now over here in between them you have sulfur bonds. You have what? Sulfur bonds. Here also you will be having one, but in between them you would have many. Okay, you can have many, my dear children, and they also have intersulfide bonds. Okay, in between them also is there, among them also sulfur bonds are there. Fine. Now what? Now what? Basically this is an antibody, and I told you antibody are having variable region. I will mark the variable region also, and the constant regions, right? The variable region is different in different antibodies. How many antibodies do you have? You have G, A, M, E, D. Gamed, I remember like that. IgG, IgA, IgM, IgE, IgD. So everyone will have different regions like this, and these are antibodies. They have to bind to what? Antigens. So you should be having antigen binding sites over here. So these are what, children? Antigen binding sites. Mention that as well. Now this is a protein. Immunoglobulin is basically antibody is a protein. Proteins are made up of amino acids, and amino acid have two terminals. One is N terminal and the other one is carboxyl terminal. So over here N terminal is here, and this is the C terminal. This is the N terminal, this is the C terminal. This is the structure of antibody. Draw it properly. Listen children, even though I said that I am a failed Picasso, it's not like that. When I have time, when the situation is not like this, okay, when I have a proper paper, I can draw properly, okay. So definitely it's not like if we try, we won't be able to do it, okay. What did I say? So basically if we try, we would be able to do it. Think like that and practice it, okay. Now the next picture which is important is this, replication of retrovirus. Now you might think this picture might not have come. It has come. Okay, this is also important, so you have to draw and practice. Draw the schematic representation of replication of retrovirus. This is the question that has come. So how do you draw it? This I don't think I have to draw it and show you, but I will tell you what is important, okay. So you make a retrovirus over here, okay. Retrovirus will be having its own RNA. So this is the RNA. So viral RNA core is there. Virus infect the normal cell. So this whole thing that you see is a normal cell. This virus is infecting. What is it doing, children? So virus has a viral protein coat. It is known as capsid. Viral protein coat is there, in which the RNA is mentioned, right? So now this RNA gets inside, the protein coat remains there only. RNA gets inside, and also certain enzymes also which are there in this particular virus, they also get inside. One such enzyme is reverse transcriptase. One such enzyme is reverse transcriptase, my dear children. Okay, what is it going to do? The viral DNA is produced by, so this is an RNA, and we have what? We have DNA. We have DNA as a genetic material. So virus feels, if I want to be within them, I should be like them. I should think of something. So it thinks of something and uses its enzyme, that is reverse transcriptase, which is going to reverse the process of transcription. In transcription, what was happening? From the DNA, an RNA is made. But over here, with the help of an RNA, a DNA will be made, my dear children. So viral DNA is produced by reverse transcriptase. Fine. Now the viral DNA incorporates into the host genome. This DNA which is going to be made, it is going to get incorporated into the host genome, that is our cells nucleus, is our DNA. Now the new viral RNA is produced by the infected cell. What happens basically? It is going to use our machinery. It is going to use our machinery. Our enzymes are being used to make more copies of it, or basically make protein out of it. It goes transcription, translation, everything. And it also forms its own RNA as well. So it has formed its own RNA. In our body, transcription happens, right? From DNA, RNA is made. So here the DNA that it incorporated, it gives rise to more RNA, and this RNA also gives rise to more proteins, and these proteins are the one that covered it, right? So all of them they get covered, and basically finally they are going to get released and they infect the other cells as well. Please remember, the first cell that it affects is known as macrophage. Macrophage is known as the factory. Okay, the factory of HIV. It comes inside and makes lots of HIV viruses in there and goes and affect other cells as well. Okay, that is how it works. So yes, that is important. So please know the diagram really well. I'm going to put a star mark up here as well. Cool. Now the next, human health and diseases. Over here you have certain structures of the drugs. What is this? Structure of morphine on 142. This is datura. And this is what? Structure of cannabinoid, page number 143. This is opium poppy. So how does the question come? You have to identify. So please remember, the one that has rails like this, okay, it is what? Cannabinoid. That means marijuana or whatever. It's not specifically given in NCERT, okay, which is why I'm not teaching you. But still I'm telling you, the marijuana, ganja, everything comes under cannabinoid category. So this is about that. Structure of morphine, as in you're talking about an opioid over here. So it looks like an O. If you see, it looks like an O. You can think like that. It is structure of morphine. Now, opium poppy, these things are known as pods. Okay, what do you call them? Pods. So you get milk of opium poppy or the latex of the opium poppy from this particular plant. So you have to identify it. The picture might be given and you might be asked to identify it. So please know what is it. Okay. Now this is what? Datura. Datura is also something that is having hallucinogenic property. Okay, it gives hallucination. So again, something that has a structure like this is datura, my dear children, a U-shaped structure. What is it? U-shaped structure. Okay, cool, datura. That's all from human health and diseases, and now we have sexual reproduction and flowering plants. This will be the last chapter that we are going to discuss today. Okay, and then the next video will be given on Sunday. Fine. I'm so sorry for the disturbance. Actually, there are so many things happening up over here. So they want to copy videos and go and things like this and that, which is why the disturbances are occurring. I'm so sorry for that. Okay. Now, up over here, you have the first and the foremost thing, that is transverse section of anther, given on page number 6, my dear children. This structure is really important. You have to study this. Okay, now I'm going to teach you that as well right now. So over here, I understand it might be difficult for few. So what do? Talking about anther, right? So anther, you can make something like this. Since I cannot see the proportion, yeah, anyways, works. So in the middle, you have certain cells which forms the sporogenous tissue. What does it form, my dear children? It forms the sporogenous tissue. We are going to label it. Let's label it. Sporogenous tissue. Each cell of this particular tissue has the ability to give rise to what? The male gametophyte, that is pollen grains. Okay, it is not the pollen grains, it is going to give rise to pollen grains. Now, this guy is really important, so it has to be protected. It has to be nourished first of all, right? So, who provides the nourishment, my dear children? Nourishment is provided by the first layer up over here, which is known as tapetum. Which is known as tapetum. Okay, now going ahead, you have two external layers. Let me make one over here. This is the epidermis. Okay, and then you have one more. This is the endothecium. Cool. Now, epidermis, endothecium and tapetum, these are the layers. In between them also you have certain cells. This is what, my dear children? This is the middle layer. This is the middle layer. So there are 3 layers or basically 4 layers up over here and sporogenous tissue, which is going to give rise to what? The male gametophyte, that is pollen grain. This is the thing that you have to draw and show. So it's just similar to it. I'll just zoom it in. Make the cells like this, that is rectangular way. They do not have a particular shape, but mostly they are rectangular, or basically you can say kind of this. I'll show you. Okay, when you are drawing you cannot zoom it in, that's the problem. So basically if this is a cell, this is a cell, you make a curve like this. Okay, that's how you draw it. But I hope you understood the funda at least. Okay. So sorry. Yeah, epidermis is there, then endothecium is there, then you have middle layer. You have 2 separate layers, middle layers only, and then this particular layer that you see is tapetum. This is what? Tapetum. Inside the tapetum you have this porogenous tissue having the microspore mother cells. Each of them can give rise to pollen grains, my dear children. Okay. The tapetum is very unique. It has dense cytoplasm and multiple nucleus, even though the picture is not given, but it has multiple nucleus and a dense cytoplasm. This is the transverse section of anther, which is given on page number 6. Cool. Now this you have to draw. This comes in drawing, which is why I put a star mark over there. Now over here you have structure of a pollen grain, page number 7. So in that, vegetative cell and generative cell is given. You might be asked the structure of the nucleus. Okay, the structure of the nucleus, which cell is bigger, which cell is smaller. So you should know vegetative cell is bigger and the generative cell is smaller. Okay, this is how the question comes. Structure of megasporangia, even though it is not the exact structure. Okay, megasporangia is talking about ovule, the structure of the ovule. I can show you the structure of the ovule. This is anatropous ovule, as in ulta. Okay, now wait. Now, okay, so say the structure of the ovule. So over here I have to make the female okay. You have the placenta over here. Okay, placenta is going to provide nourishment to our little guy, that is the ovule. Fine. Fine. And it is going to provide nourishment with the help of something like this, which is known as funicle. And the point of attachment is hilum. Cool. The ovule which is there, it has 2 integuments. One is this and the other one is this. This is outer integument, okay, and the other one is inner integument. Cool. This particular region that you see, it is known as micropylar end, or this is the micropyle region, and the opposite end it is known as chalazal end. Okay, here also you have the cells of the nucellus. What do you have, children? It is not given in your NCERT, but yes, there are so many cells filled up here. And what do you call this as? These cells as nucellus. Works? So that is the structure of the megasporangium with lots and lots of cells up over here. Not all of them are going to give rise to the female gametophyte. Only one cell is going to give rise to what? The female gametophyte. Okay. So they have given anatropous ovule as in the ulta ovule. Over here you see the micropylar end is here, chalazal end is here, but the funicle is up over here. So instead of having the funicle up over here, the funicle is there. So the placenta might be here somewhere. Okay, so this is inverted, or basically what do you call it as? It is what? In anatropous ovule. Page number 9. Now again a picture is given over here. the same picture where the anatropous ovule was there. Listen to me very carefully. This picture would give you questions. Okay, what type of questions? It can ask you, in which plant you find multiple carpels or syncarpous pistil? Multicarpellary or syncarpous pistil. What do you mean by multiple or multicarpellary? Basically, it has multiple carpels or multiple female reproductive things. So, you see over here in the case of bee, it has so many, all of them basically, it is having multiple carpus, okay, and it is also known as syncarpous. Why is it known as syncarpous? Because all the carpus are fused together. They are what, children? Fused together, which is why it is known as syncarpous. But in what plant do you see this? You see it in papaware. Over here, I will zoom it and show you. Over here, you see them in papaware. Can you distinctly identify there are so many separate carpels over here? No. Why cannot you? Because it is syncarpous. It is in joint stage, and this is shown by papaware. Okay, this is shown by papaware plant. That is an example of this. Now, a question can come, in which plant do you see apocarpus? Okay, apocarpus condition. Apocarpus means basically they would be, the carpels will be separate. Here also you will be having multiple carpels, but it will be separate. So over here you see they have apocarpus condition. So many carpels are there. Now, which is this plant? Its name is Michelia. Its name is what? Michelia. Again, the diagram, the name is given up over here. So you can get questions like this. What do? You put a star mark up over here, because saying that this is important. You don't have to draw the picture, but this is important. But in this picture's case, you have to draw it sometimes. So this is why you put it star. Cool. Chalo. Next thing, important for 1 marker question. Okay, that is what I said. Now, next thing, you have the picture of the embryo sac. Okay, embryo sac, the female gametophyte over here. So this picture is very much important. Antipodal cells in the chalazial end, okay, filiform apparatus and everything in the micropilar end. You have synergids, you have an egg cell, you have 2 polar nuclei. This whole thing that you see is the central cell. If you want a proper picture, I can show it to you. Okay, I can show it to you. I'll show it to you just a second. Okay, so clistogamous and chasmogamous flowers of Viola, Oxalis, Commelina is given up over here. So you should know that these flowers that is being shown up here, it is what? Clistogamous. Clistogamous are always towards the lower area. Clistogamous are the closed flowers. Closed, you can remember like that. And the other flowers are chasmogamous flowers. This plant is Oxalis or whatever, Viola, Commelina, whatever plant is there. All of them basically have both these flowers. Now, again, I'll give you this picture and tell you what type of pollination is happening up over here. So this is a bee, and this is basically the orchid which is there. So this is insect pollination that is being shown up over here. So that is how question comes over. So you don't have to draw any of them. So just put a tick, because you know it is important. Question can come from this, so that you have a memory of it. Now, what can come to draw? Mature embryo sac, and that is what I am going to show you up here. So mature embryo sac, it looks like this, my dear children, where you have to form something like this. Okay, now I am going to make the micropilar end that side. So here you have synergids having the filiform apparatus with the nucleus. Okay, now over here another cell, that is the egg cell, again with the nucleus. Over here you have 3 antipodal cells. Over here you have 2 polar nuclei. That is what you mean by the mature embryo sac. Okay, so this is what? The egg cell. This is what? Synergids. These are what? Polar nuclei. This is what? The central cell, the biggest cell over here. These are what? The antipodals. This is which end? Chalazial end. This is which end? Micropilar end. Remember, micropilar end had an opening through which the pollen tube enters and basically fuses with this. This is the embryo sac, and this is what you have to draw. Okay? Cool. Now the next thing, you have typical dicot plants embryo, okay, page number 19. LS of embryo of grass, that is monocots. This is dicot, this is monocot, page number 19 again. And this picture you don't have to draw, okay. This and this can come for drawing, this won't be coming for drawing, you have to label it. So you should be knowing that this is coleoptile, this is plumule, this is radicle, and this is coleorhiza. And this extra thing towards one side is scutellum. And this is the endosperm. What is this particular thing? This is a monocot seed. This is a structure of a monocot seed, my dear children. For I'm making you understand this, I can draw something and I can show it to you. Okay, let's make the dicot ones. Okay, dicots have two cotyledons. Dicot have two cotyledons, and cotyledons is something that stores food, my dear children. So basically, there's an axis over here, and this axis looks like this. Over here, okay, wait, instead of drawing like this, I can show you the other image. Okay, first I have to show like this only, then only you will understand. So make the embryonal axis. This is not for you to make, okay, this is for you to understand clearly. So children, you have two cotyledons like this. Okay, cotyledons are there for storage of what's important nutrients, because there's a growing embryo. This thing that you see is the embryonal axis. This is the plumule, which is going to form what? The future shoot. Okay, it is going to form the shoot. This is the radicle, which is going to form the root. There is a root cap as well. There is a root cap as well. This region is hypocotyl and this is epicotyl, that is there. Remember, always above the embryonal axis, or basically above the level of the cotyledon attachment, there will be plumule region. Below this level, you have the radicle, or basically this the lower region. Okay, radical region is something that gives rise to the roots. Now, how do you draw it according to the picture that is given in your NCERT? I am talking about dicots. It has two cotyledon, 1 and 2, right? According to the picture given in your NCERT, you draw it like this. So, you make something like this. Okay, so what did you do? Basically, these guys, it went down like this. So the one that is above the level of attachment, above the level of their attachment, what do you call this as? You call this as plumule. And this one is radicle, the lower region. It is covered by what, children? The root cap. And over here you have two cotyledons. This is the structure of typical dicot embryo. Okay, these are the two cotyledons. That's all. That is all. This thing they twisted and made like that. Okay, this is for this guy, that is the dicot ones. So this is the hypocotyl, the lower region, and this is the epicotyl, which is a termination, that is plumule. Cool. Now the root cap can be shown like this. Now LS of embryo of grass. Now this picture seems to be very complicated for me. So what do? Basically, listen to me carefully. So it also has embryonal axis like this, but as I told you, it just has one cotyledon. So let's make one cotyledon and try to understand the image first. So this is the cotyledon that it has, and its name is scutellum. Only cotyledon which is there, and it is known as scutellum, my dear children. The region above this is known as plumule. Region below, over the lowest region, is known as radicle. The radicle is covered by root cap. And I told you there is only one cotyledon. There is only one cotyledon, but that is not enough. So basically, usually when the seed is formed, it has endosperm, and endosperm is the one that gives nourishment for the zygote to form into a complete embryo. So embryo says that I don't want endosperm, I will take it and make my own reserve nutrients in the form of cotyledons. But sometimes the endosperm still persists, and that endosperm stays like this only. So this is the extra endosperm that you are having. Okay, and you have the cotyledon as well. Now what happens? What is one more difference between the dicot and monocot is that the plumule is basically surrounded by one extra layer, and this is known as coleoptile region. Over here also the radicle is also surrounded by something, and this is known as coleorhiza, my dear children. This is known as coleorhiza. So this is the basic structure, and they have made it a little complicated, and they have shown it up over here. So in the picture that is given in your NCERT, see, the plumule is there, radicle is there, coleorhiza is there to support this, coleoptile is there to cover the plumule, and scutellum, that is that is cotyledon, will be there, and little amount of endosperm is going to be there. Let's see if all of them are actually there or not. So over here you have the coleoptile, and the coleoptile which you see over here is covering what? The shoot apex, that is the plumule. Here you have radicle, the radicle has root cap, and then you have coleorhiza as well. There is scutellum, and there should be endosperm, which is not mentioned up over here, but it is mentioned up over here. So these are everything. You have scutellum towards one side, and then you have endosperm. What is pericarp? Pericarp is the outer covering of the seed. So everything is happening inside the seed, right? So outer covering of the seed is known as pericarp. Okay. Now this structure, if you want to draw, you can draw this structure as well. This is what they prefer you to draw. But this is kind of very confusing for me as well. I don't like to draw this, but let's draw. So you have to make a shark's mouth over here. I think like that. Okay, so there is a sharp's mouth over here. This is the mouth region, and it has a small bulge like this. One more mouth, which goes like this, and which comes back like this, and forms like this. Okay, I am making a smaller image. Over here you have the scutellum, and then it goes up here, comes up over here, goes to that side. From there again it is going to get connected like this. But I do not prefer this image because nothing is clear up over here, and they call this region as scutellum, they call this as the radicle, and this as the root cap, and then this as the coleorhiza. Okay, this is what the epiblast, this is epiblast, some other structure which is there. Okay, and then they have mentioned over here that this is what coleoptile, and over here they have mentioned something like this and mentioned it as shoot apex, which is completely not something that anyone could prefer to draw. Okay, and I have drawn it in a bizarre way because I preferably do not find this drawing very captivating. Okay, because this is not self-explanatory. So LS of embryo, if it is asked, you can draw the one that I have shown you, or you can draw this also. Not LS of embryo, you can draw this only. Sorry, if you are asked about the structure of the embryo, general structure of monocot seed, you can draw this. Okay, works. So it is kind of confusing, but yeah, we have to practice it. So that's all from this particular chapter, and from human reproduction and everything. We will be having one more video, my dear children. For this particular chapter, that's enough. So if you're thinking that you are ready for practicing diagrams, do let me know in the comment section. We are going to practice more and more diagrams, okay? And in human reproduction, you have lots and lots of picture that is really important. So be there with me sharp at 6 p.m. on Sunday as well. Okay, so I hope this video helped. I know that I cracked at some areas like this where I do that. Okay, it was very embarrassing, but yes, that is the truth. Okay, this picture is not that great, and I would not recommend NCERT to have it, but yes, that's how it works. Cool. So, chalo. That's all. No more wasting of your time. See you in the next video. If you're liking the video, do not forget to like, share and subscribe. Bye-bye. Take care.